Everyone, I'm Lauren Ponisio, and I'm an assistant professor at the University of Oregon. I was a postdoc at BIDS. What was it? Feels like a really long time ago, but I'm really excited to be here today, remotely, as it were, to talk about time-evolving networks and ecology. I hope to first convince you guys that networks and ecology are awesome and there's a lot to be done, and also think about the new methods that are being applied in data science. So it's been a while since we've really kind of moved past the idea that species interact one to one. There used to be these really nice stories that there were these fig trees and they were only pollinated by this one species of fig wasp. But in the end, eventually we realized that that was mostly due to incomplete data and bad sampling, and probably the idea that humans just really liked the idea that that was the case. But now we think much more about species interactions in terms of large networks of many interacting species. So here's some of my favorite examples. These are Frugivory networks, so seed dispersal networks. So different species of animals eat fruit and then they disperse the seeds in the Pantanal in Brazil. And you see there are these different modules within the network. There are some like bird modules that are larger and then smaller. There are some also large and small mammal modules. And in the very corner over there, you see the purple, there's actually a little fish module. So fruits will fall into the rivers and then these fish will disperse the seeds. We've also been able to reconstruct ancient food webs. So based on various shale formations, people have used the body size of species as well as actual fossilized evidence of a fish being maybe eaten by another fish to infer these ancient fossilized networks. Fisheries are a place where there's just incredible amounts of data because we have been monitoring fish catch for a really long time. And so people have built food webs based on body size, as well as thought about, how do humans and like, for example, fish trawling in this image fit into this larger marine food web. Near and dear to my heart are plant-pollinator bi-partite or two-party networks. So you have the plants on one side of it, and then you have the pollinators. These are mostly considered mutualisms. So the pollinators are providing a service to the plants, mainly pollination. And we've seen that incredibly diverse arrays of pollinators from bees to flies and butterflies are involved in these interactions, and which sometimes have these kind of module formations. And lastly, my all-time favorite network was based on taking Egyptian art, particularly hunting art, and they depicted the animals with such accuracy that Justin Yickel et al. Was able to actually reconstruct the animals present in different time periods based on the art. And they were able to watch the collapse of these Egyptian food webs, potentially due to overhunting, but also climate change. So just an amazing example of like bringing art to ecology to understand how communities change. But one kind of issue is that networks really are not static. And I think that, people in the social network world have probably come along a lot farther than ecologists on this one. So instead of just characterizing the network as one snapshot, people have started pushing that, these networks are changing and evolving through time. And so there's this whole world of time evolving networks, and most complex systems evolve over time. Very few of them, if you look at that Pantanal network, the year after it was probably a little bit different, and same as some species move in and out of those networks. Their structure is often represented as a time evolving network because of this. So you have a sequence of network snapshots, like the graph at time t, for some t from time period 1 all the way to whatever you have. In ecology, to be honest, we often don't have very many time points. Maybe we have 10 or 15, but probably in social networks and things like that, people have a lot more of these slices. And so when it comes to measuring how networks change through time, the first approach we can use is just to get rid of the graph. So we apply some network measure and turn our graph into a function of that graph. So examples are like the mean degree, the diameter, the clustering coefficient, what have you. And we can then use these data points as classical, bits in a time series. Pros of this is it's super easy. There's lots of existing tools. I'm throwing myself onto the bus right here. We've done it before. We were looking at time since assembly in these hedgerow communities, which you guys hear more about. And we just looked at various network metrics and how they changed. So it was quite simple to do. But the cons is it discards basically all the network information, and it's really not the best for being able to make inferences about time evolving networks, just because we've lost so much information by collapsing everything down into this function of the network. So a better approach might be something like actually fitting some kind of generative model to the graph and then looking at those change in parameters through time. So we can fit some kind of generative model. We can choose some theta at time t and generate a graph by drawing a graph sequence. And we can observe how g of t, we can observe g of t and infer the best theta at time t. I'm going to give you guys an example of this as well using real data. So the pros of this is we can directly model the entire time varying draft. It's not just collapsing it down into some network metrics. It naturally generalizes to time series techniques. There are lots of powerful methods for deciding how, when, and if structure changes. There are also powerful methods for coarse graining how structure evolves. And it is a rich and unexplored domain. There's lots to be done here. So lots of great methods to develop and those kind of things. But the cons are the math is a lot more difficult. And it's really hard to explain to people. It's a lot easier to think about how the average degree of a network changes through time, but it's a little more abstract to think of like how the theta of the graph changes. But regardless, we've given it a go, and I wanted to show you an example of this. So in the Northern Central Valley, which you guys maybe have interacted with, out in Yolo County, there are these hedgerows. So they're native plant restorations. They're planted alongside agricultural fields with the idea of promoting pollinator restoration as well as promoting pollination of those crops. And so here are a couple, like a slice of what those networks look like. In the green dots are plants. They're weighted by how many things are visiting them, and the yellow dots are pollinators. And we collected many, many bees from this area. In the end, we ended up with about 10 years of data, over 500 sampling hours, over 130 different bee species. So incredibly rich ecological data set. And what we were able to do is fit that generative model. And in this case, we chose the generalized hierarchical random graph model. It basically ended up fitting a dendrogram to our network. We then inferred two versions of that model. One where the change of network structure, one where there was a change in the network structure parameters between two time points, and one there was no change in network parameters. And we were interested in, as these networks are assembling through time and changing over this 10-year period, is it basically the same graph over and over and over again, or are there major reorganizations or change points in the structure of these networks? We then used Bayes factors to choose which model, change point or no change point, fit the data better. And so what it ended up looking like is here are some illustrations of what the models what the networks look like. And the data suggested there were multiple change points in these hedgerow networks. Here is an example of one. And so we had one hedgerow where the network in years 1 to 3 were not distinguishable from each other. Then there was a major change point, and then there was the network in years 4 to 7, then another major change point, and the years 7 to 10. And we saw these change points occurred a lot more in the assembling communities than communities that were more mature. So we also sampled hedgerows that were really quite old and very stable. And so those tended to not have very many change points, whereas these assembling communities did. So this really suggests there's something really dynamic going on, and species are being added to the system and then leaving. And what this meant is that, when we fit those hierarchical random graph models and we looked at our parameters, which, theta hypothetically, but in this case, we were looking at the parameters of beta distribution. The graph model in the first, series of time points was not the same as the second, was not the same as the third. And when we look at the species, you see that species are really dynamically moving in and out of various modules in the network. So the dark red module is that like center, highly connected module in the middle. And you have these species, some that just kind of stick around in there and hang out. But then you have these other species that are just blipping all over the place and actually causing these major change points. And this was really interesting to us. We were wondering, who are these little guys? Like, what are they up to? Or how are they changing their interactions? And particularly, like, how do they contrast with the species that are just staying in the middle of that network over and over and over again and not really moving around? If you guys are interested, all the code is available on GitHub. It's been a while though, so it's actually in Python 1, if you can believe it. But I'm always trying to get Lido Peel, who is the original creator of the change point analysis, to update his code. But it's all out there. Check out GitHub if you'd like to see. What's next then? We have these maybe large scale changes in network patterns. We weren't really able to tie the change points to anything meaningful in terms of what was going on in the world. It seems like they were happening in the life of these various hedgerows at somewhat random points, and that a variety of species were driving them. And so we really wanted to like zoom in and ask, what are the node level network dynamics? How are individual species, in this case bees, changing their interaction patterns through time? And to do this, we kind of went back to the whole, let's just summarize this roles of species by taking a certain metric of their interactions in a variety of years. So say we have year 1 and year 2 of a certain network, and we have a variety of bees interacting with plants in green. And let's focus in on this little red bee. And so in the first year, she interacts with 2 different species of plants, so something kind of daisy and some kind of penstemon. And the next year, she interacts with the lupin. And so here we show that there's this variability in her partners. And we can take all of those species she interacts with and array them in an n-dimensional space and plot out the variability of her interactions with these different partners through time. Like something like partner beta diversity, if you guys are familiar with the concept of beta diversity. So partner turnover or partner variability. And we can do the same for another species that maybe did not change their partners as much and therefore has a much tighter area in this n-dimensional space of interaction partners. We can then take the coefficient of variation of these distances to the centroid or to the middle of that space and use that to quantify how variable you are in your partners. This is really commonly done in ecology when we think about species composition at various sites. So in this case, the species or the species are the sites and their partners are like the species in traditional ecology beta diversity. Maybe that was an overly complex way to explain that. But the other way we can quantify role variability is to think about network motifs. So people have thought about motifs as the building blocks of networks. You can take a network and break it down into these fundamental little quantifiable pieces, and they can vary based on how many nodes are involved in them. So there are 3 node motifs as well as 4 node motifs. And we can ask, different species may be involved in these different motifs throughout a time period. So for example, we have these 2 years again. We have year 1 and year 2, and we're focusing in on 2 species, A and B. They're present in both years, and we can look at their involvement in a variety of different motifs. So on the different numbers 3, 7, 33, 49 are all different motifs that we can break a network down into. And you see that first year, A1 is involved in motif number 7 20 times, but none of the others. And then in the next year, she's actually involved in motif 3 twice and motif 7 only 4 times. And so we can see this variability in kind of the niche or the role that a species might occupy in a network. And so we wanted to take those metrics and ask whether they actually meant something for species when we think about their occupancy or their persistence across a landscape. And to do this, we used an occupancy model. So this kind of treats different habitats like patches. So we were thinking about the hedgerows again. So we have these like isolated hedgerows in this agricultural landscape. And we wanted to ask, okay, you can either be present, z equals 1, or absent, z equals 0 in the initial year. And then you can either colonize to be, if you were originally absent, to become, to occupy that space, or you can persist, or you can not colonize. So that's, you were absent in that year and you stayed absent in the next year, or you can go extinct. And all of these possibilities together can get us whether a species is present at a certain patch occupying that landscape. And we wanted to ask, how does interaction flexibility in terms of partner variability and this motif kind of positional variability affect a species' ability to colonize and persist in a landscape? So this is in the hedgerow system. We looked at how the various population parameters varied based on a species' partner variability and role variability. And what we found was that partner variability actually had a significant negative effect on colonization. So species that were very variable in their partners were less likely to colonize other patches, which is not what we predicted. Interestingly, however, species that were very variable in their roles were better able to persist in the landscape. So in general, partner switching led to lower colonization, which actually led to lower equilibrium occupancy, or the number of patches species occupied, whereas role switching led to higher persistence and actually higher equilibrium occupancy. We were a little bit stumped as to why partner switching might lower colonization. We thought one thing is that potentially in this really resource scarce landscape, being able to switch your partners a lot might not actually be a great thing, like that you're using really not very nutritious resources, which are plants. And in the long run, even though you're visiting lots of things, you're not really getting what you need. And so you kind of end up staying in certain patches. Whereas it made a lot of sense to us that the ability to switch your network role to occupy different positions in the network would help you persist through various years, and that would result in a higher occupancy. So you'd be occupying more patches in this landscape. Another example of this was from Yosemite National Park. So we did basically exactly the same thing, but we were particularly interested instead on how interaction flexibility might promote persistence through an extreme drought. So this ability to like kind of resist perturbations or change. And so we were working in Yosemite National Park. It's an incredibly beautiful place. There has been a really long history of allowing natural fires that are created by lightning to burn. It's pretty deep in the backcountry. A lot of Scott Stevens' lab works in this area. It's called the Illilouette Basin. We went out and we sampled pollinators through this rather extreme drought. That was not the intention of the project, but we had this crazy drought happen kind of in the middle of it, particularly in 2014, where it just did not rain at all. And so we had these data points before the drought when populations were doing well. And then we had this huge perturbation to the system, this drought, which really knocked back plant populations. And we wanted to ask, does the ability to be a flexible interactor help you resist this extreme disturbance? And so on the y-axis, we looked at the log ratio of abundance of these pollinator populations. We would expect that if they were able to resist the drought, their log ratio would be right around 1. And on the x-axis, we have biodiversity. So this was the diversity of fire histories in the area. I mentioned that this area has all these beautiful, amazing lightning strike fires. There's all this heterogeneity of habitat. We want to know, does all that heterogeneity of habitat help you resist this perturbation? As you can see by the flat dotted line, we did not find that. So in general, species after the drought had lower population sizes. Their abundance decreased. That's why the line is below the 1 marker. And it did not respond to the pyrodiversity or fire diversity in the area. However, once we incorporated partner variability, so this is again that kind of partner beta diversity or the ability to switch your partners, you can see that there is a significant interaction. So with more diversity of fire histories in the area, in addition to the ability to switch your partners, this enabled you to kind of like take advantage of all that awesome heterogeneity and resist the drought perturbation. We found something really similar with role variability. There wasn't as much role variability in this landscape for some reason. Species tended to stick more within the same motifs over and over again, but we did see the same significant positive interaction. So if you were more variable in your role, you were better able to resist the drought. So what's next? We've talked about this, the larger network and its change through time. Then the variability in the nodes, and this is kind of like summing up the individual interactions from lots of different individuals to the species level. And next, we really want to zoom into these individual level networks, so the actual individuals within a species. For example, we have this gorgeous bumblebee, and you can see that giant pollen ball on her leg. And we can actually pop those off. So we catch them, we put them in little tubes, we kill them. And then we just use a little dissecting tweezers to pull these little pollen balls off. And then we can use next-gen sequencing to go back in time and actually reconstruct all the plants that she visited before we captured and killed her. But an incredible amount of information there. So we can build a network not with just all of these individuals making up a species, but at the individual level within a species, in between species. We can also pull out her gut. We can dissect out her gut and look at her microbiome, her bacterial microbiome. That's using 16S primers, a standard kind of technique in Illumina sequencing. And we can screen for specific gut parasites, which are known to affect bees. So these are a variety of fungal and trypanosomatid parasites that cause a lot of problems in bees. And all of these things will actually be in their gut as well. And so we were looking at a system where we had the data for over 1,000 bees on their pollen, their microbiomes, and their pathogens. And this is up in Northern Central Valley again. And we predicted that intraspecific or within a species variability in floral or pollen interactions would be more variable than parasite and microbe interactions. We assumed that microbes and parasites, they have a little more specificity. They need to be able to infect the host. In terms of the microbe interactions, they need, the host kind of needs to let them kind of hang out there without their immune system attacking them. And we thought, if you're a free living bee just wandering around, you'd be more likely to visit various pollen species and things like that. We also thought that variability in the microbial interactions would be lower when the plant-bee interaction networks were dominated by a few highly visited hub species. If you remember the networks I presented talking about the chains point analyses, sometimes in pollinator networks, we see these highly attractive plants, particularly in agricultural systems like sunflower or brassica, these things that offer lots of pollen and lots of nectar. So lots of species are visiting them. And because, and this is a key point, bees actually pick up their microbes and parasites from flowers, we thought that, okay, if all the bees are only interacting with one kind of flower, we'd expect less variability. These are still pretty early stages and we have to kind of check on some stuff. We did not see this though. We actually saw huge variability in microbial interactions as well as parasitic interactions. And we're still looking into that, much more so than the pollen interactions. And we did see some evidence that the interactions and the microbial interactions were less variable when there was this kind of host, this core kind of peripheral structure to the network, but we're still looking into that. If you guys are interested in these methods, please check out, there's a couple packages in R, particularly in this StatNet arena, there's a network dynamic as well as a couple others. In Python, there's a really neat package called DynetX, which has some really neat tools. A lot of them are focused on visualization and not so much analyses. I think those tools are still under development. And with that, I really want to thank all of you for listening to my talk and hearing a little bit about ecology and bees. Hopefully it was just the right amount of fun bee facts to get you excited about all the cool uses of networks in ecology. And also think about how, maybe in your field, these temporal or time, evolving networks might be applied. And with that, I like to really thank my lab as well. A lot of this very quickly, actually, once you become a professor, it's like you become really outdated. I thought it would take longer, but a lot of this work was done by my amazing postdocs and lab manager. And so with that, I'd love to take any questions when we actually have the opportunity to interact in the workshop, but here are some main results. We're trying to move past summarizing networks with metrics to fitting time-dependent generative models to examine dynamics. Node level analyses are still in the early times. We're just kind of back in that like summarizing the networks with metrics world, but we've got some promising results that even at that level, we're able to show that this node level variability has ecological implications. And soon we're going to have some more individual level data due to all of these incredible new sequencing technologies that are coming out, and hopefully we'll be able to examine these individual level networks across many different types of interactions. Thank you.